Now, lipoproteins actually communicate while they're in the circulation. So let's take a look at this level of communication. Let's start out with our main lipoproteins, those being the chylomicrons, constructed of dietary lipids and cholesterol. These, of course, have core protein of ApoB48. Our other lipoprotein, which carries fat and cholesterol, VLDL, which is synthesized by hepatocytes, and this is consisting of core protein ApoB100. Now these also, of course, consist of triglycerides, cholesterol esters, and phospholipids. So when these are secreted by intestinal cells or by hepatocytes, this is the components that are part of these lipoprotein particles. It consists of the core protein that adds the structure and scaffolding for the lipoprotein, and then the major components of the lipoproteins, being the fats. Now, we know that ApoE is involved in recognition of these particles for the HSPG. So recall the HSPG, this is the membrane of the endothelial cell. The HSPG is a transmembrane protein, core protein here, which faces the lumen of the blood vessel. ApoE is an apoprotein that is an accessory protein that binds to either the chylomicron or VLDL. Now, of course, the lipoprotein lipase, LPL, is the lipase secreted by the extrahepatic tissue. And these, of course, are the metabolically active tissue, such as skeletal or cardiac muscle, or adipose. In other words, tissues that have a high demand for fatty acids will synthesize and secrete LPL. So to prevent these factors, LPL, from washing downstream, it is bound to this heparan sulfate proteoglycan on the surface of endothelia to maintain this lipase activity within the circulation of the tissue that needs the fatty acids. The lipoprotein itself has ApoE, which also holds it to this HSPG. So now we bring the enzyme and the substrate closer together. Another factor besides ApoE is ApoC2. This is also brought to the binding of LPL on the HSPG by the lipoprotein. This is an activator of lipoprotein lipase. So thus the ApoE factor will localize the lipoprotein to the HSPG, which keeps it close to the enzyme bound, and it also delivers the activator for the lipase. Now, where do ApoE and ApoC2 come from? Recall that I mentioned that the chylomicrons synthesized by intestine and VLDL by the liver, when they're secreted, they are consisting of ApoB148 and ApoB100, their core proteins, as well as the fats that they're carrying. The ApoE and ApoC2 are loaded onto the lipoproteins after that they arrive in the circulation. And this is done by transference of these factors from HDL, high-density lipoprotein. So HDL essentially serves as a reservoir for these apoprotein factors. So when the HDL encounters a lipoprotein with just its core protein, it will then transfer the ApoE as well as ApoC2 to the lipoprotein, which now functions to attach it, adhere to the HSPG, localize to the lipase, and deliver the activation protein for lipoprotein lipase. Once the chylomicrons and VLDL are processed to their respective small products, chylomicron remnants, or LDL, portion of the ApoC2 and ApoE from the chylomicron remnants is shed and released and bound back to HDL to serve again as this reservoir of these factors. And LDL also returns these factors. This, in a sense, allows chylomicrons to bind to HSPG through the small last remaining ApoE that's associated with it, or to LRP on the hepatocyte, and LDL can bind to the LDL receptor through its recognition sequence on ApoB100. Now the other communication that goes on between lipoproteins is between VLDL and HDL. In this case, HDL is able to transfer cholesterol esters from HDL to VLDL. In return, VLDL is able to transfer triglycerides from VLDL to HDL. This is performed by the enzyme CETP for cholesterol ester transfer protein. Now the purpose of this is to maintain a cholesterol reservoir in the body. Recall how expensive energy-wise cholesterol is to synthesize, and so the body finds a way to minimize its loss of cholesterol. So HDL is involved, of course, in reverse cholesterol transport, and its job is to rid a cell of extra cholesterol and deliver this back to the liver. However, because cholesterol is so important to cell function, HDL is going to transfer some of its cholesterol esters to VLDL. This will allow VLDL processing to LDL, which is our cholesterol-rich particle, and then this allows cells another round of potential uptake so that they can maintain their cholesterol needs. Now, the reverse cholesterol transport from HDL occurs when cells have taken up sufficient cholesterol and this has been done by the LDL receptor family, the LDL receptor. And if sufficient cholesterol has been taken up for cells and we have excess cholesterol, this cholesterol can now be removed from the cell. And this is the job of HDL. HDL will pick up this extra cholesterol, ultimately delivering it to hepatocytes. Now this process is going to involve a very important enzyme called LCAT. And LCAT stands for lecithin cholesterol acyl transferase. This LCAT enzyme is associated with the HDL particle, and it's solving an important chemistry problem. Cholesterol is in the plasma membrane of cells. It's in a hydrophobic environment, and it likes that environment. So to remove cholesterol from this environment takes an enormous amount of energy. The cholesterol in HDL is also in the core of the protein, solubilized in the fatty center, the hydrophobic center. And so if we just had cholesterol in the HDL and cholesterol in the membrane, it would easily be reversible, because the free energy change between these two would be approximately 0. So in order to ensure that this reaction doesn't go backwards, the function of LCAT is to make a cholesterol ester. So HDL is going to take the cholesterol from the cell, attach a fatty acid to it, making a cholesterol ester, and then the association, the solubilization of the cholesterol ester forming bonds inside the HDL particle is much greater than the cholesterol forming bonds inside the cell itself. So that will ensure a one-way direction of cholesterol movement from cells to HDL. Now once the HDL becomes loaded with cholesterol, it enters the hepatocyte. It does so by a receptor called the scavenger receptor. Then this is unloaded. The cholesterol esters are removed. The HDL returns to the circulation to continue its activity in reverse cholesterol transport. The cholesterol esters that are removed from the HDL and the hepatocytes now, of course, have two fates, one of which is for repackaging in VLDL, which is then placed back into the circulation, or for disposal as bile salt synthesis. So this pathway of lipoprotein communication is important for the factors that are being held in a reservoir in HDL, such as ApoE and Apo2C, from being transferred onto the lipoproteins that are the fatty acid and cholesterol carriers so that they may be processed. Fats removed and delivered to extrahepatic tissue. The cholesterol, because it's a very expensive molecule to make, if we take our Western diet out of the situation where we have high cholesterol in our diets, cholesterol is to be conserved in the body because we don't want to go through extra expense to make it when we can conserve it. So the conservation is twofold. First, we're going to add these cholesterol esters that have been scavenged from cells, that are waste products essentially from cells, put it back into VLDL in the circulation. This will allow cells to have essentially a second chance at obtaining cholesterol if their needs call for it. The second method of conserving cholesterol is in bile salts. Recall that bile salts can be taken up by the enterohepatic circulation. These are taken up in the terminal ileum, returned through the portal circulation to the hepatocytes, and then reused for bile, for solubilization in the bile. Recall that approximately 95% of the bile salts are recirculated. So this will maintain our bile salt pool and prevent any excess cholesterol from entering the bile salt synthesis pathway, thus conserving our cholesterol.